This video is about introduction to the cell, taking a look at prokaryotic, eukaryotic, why cells are so small, basic function of the cell organelles, and some difference between plants and animal cells. We'll also take a look at some examples of AP questions that might be asked of you as far as the cell parts and functions. So the first thing we're going to start with is classification of cells, and that's going to be prokaryotic and eukaryotic cells. And you're not going to be given a picture to label as far as a prokaryotic cell and a eukaryotic cell as far as the parts, but just kind of comparing, contrasting what the difference is. You may be given a picture that has the cell parts labeled on it, but then you're going to need to know the function and kind of how they're related. So when we take a look at a eukaryotic cell, this is probably the typical cell that we think about. Eukaryotic cell means it's going to have a nucleus, and it's going to have a nucleolus present. It's going to have membrane-bound organelles. If it's an animal cell, it's just going to have a cell membrane around the outside. If it's a plant cell, it would have a cell wall around the outside. So a eukaryotic cell is the typical cell that we think about when we think about cells. Prokaryotic cell is going to be a bacteria. It's going to be a cell that does not have a nucleus. It is not going to have membrane-bound organelles, although I will highlight, and this is an important part, that they are both going to have ribosomes. So ribosomes are for protein synthesis. And so both prokaryotic cells, like bacteria, and eukaryotic cells, like plant and animal cells, are going to have ribosomes. So that's something that they're going to have in common. So, just recognize a typical prokaryotic cell. There's no nucleus. The genetic material is just kind of in a location. It's not enclosed in a nucleus within that cell. So I found a little chart here that I liked. Not sure who the little person is there. Maybe that's Dora the Explorer, but anyway, it does a good job of kind of comparing prokaryotic and eukaryotic cells. And I highlighted what I think is probably the most important, and I think you should certainly have the definition that prokaryotic cells are not going to have a nucleus, and eukaryotic cells are going to have a nucleus. And as I said, no membrane-bound organelles in the prokaryotic or the bacteria, but we are going to have membrane-bound organelles like the mitochondria and other organelles found in the eukaryotic cell. What they do have in common, and they just give it a different name. They say the 70 or the 80S, but just keep in mind that they're both going to have ribosomes for protein synthesis. And reproduction, bacteria are just going to split. Eukaryotic cells are going to undergo mitosis. It's going to be splitting, but they have two ways to split. You can produce new cells by mitosis, where you get exactly the same genetic information, or you can produce by meiosis. Prokaryotic cells, binary fission, is very similar to mitosis, although we know that bacteria can mutate. So there is some level of mutation and evolution going on with bacteria. Average size, much smaller for bacteria, prokaryotic cells. They can fit inside of a eukaryotic cell. So when you take a look at the organelles, this is not to scale. This cell could fit inside here, and it might be like the size of the mitochondria. So that is a comparison of prokaryotic, eukaryotic cells. General description of what they have in common and what's going to be different. We're going to move on to the plasma membrane, and we're going to spend time taking a look at the functions of the plasma membrane because there is going to be more than one function. So in our study, in the next unit here, we're going to talk about transport, we're going to talk about enzymatic activity when we talk about cellular respiration and photosynthesis, and we're going to be talking about transport. We're also going to talk about cell communication. So there's a number of different functions of the plasma membrane. So I'll just go through those. Selectively permeable, which means it can allow some substances in, some substances out. Basically, it's going to be based on size, charge, and whether it's hydrophobic or hydrophilic. It contains transport proteins to move materials across the membrane, and we'll see a picture on the next page. So that if it's too large or does not have the right charge or the polarity, we can still move large molecules across that membrane. Enzymatic activity, so the enzymes for photosynthesis and cell respiration are actually going to be located in the plasma membrane. Cell communication. Membrane proteins have specific binding sites to combine with signaling molecules. So that is a topic that we'll talk about. Epinephrine, we'll talk about steroids, and how they can communicate with various cells and get a response. And then cell-to-cell recognition. There's carbohydrate chains that are going to be attached to the membrane proteins to identify cells and cell types. So this study is really about taking organs and genetically modifying them. So let's say in animals, instead of having the genetic identification for like a pig heart, you would have the genetic identification for a human heart. And so part of what happens is these carbohydrates that identify a cell, if you are doing a heart transplant and you don't have a human heart, we're working on right now, or scientists are working on right now, being able to genetically modify the pig heart so the carbohydrate chain doesn't identify it as a pig heart, it identifies it as a human heart. So as far as organ donation and transplants, that's something that's being worked on. So on this sheet, I've kind of highlighted some important parts here. Carbohydrate chain on that protein molecule is going to identify the cell and cell type. The channel protein is going to allow substances in and out. You can see there's an opening there that can allow large substances in and out. And then we have these peripheral proteins, and we have the integral proteins. Integral proteins just means they're integrated or within that plasma membrane, and they can be important for cell communication or enzymatic function. So again, we're going to take a more in-depth look at the functions of the cell membrane. Next question is, and deals with the cell membrane, is why are cells so small? And when we take a look, and you may have remembered doing an activity in your previous biology class, there's a couple activities that I know are done. One of them is just cutting out models, paper models of different sized cells. And so in this, you're just going to kind of compare the surface area, the volume, and then the surface area to volume ratio. And you can see, obviously a large cell is going to have the largest surface area, it's going to have the largest volume. But the important part here is the surface area to volume ratio. And as you can see, as you get smaller, what happens to that surface area to volume ratio is it's going to increase. So for the size of this cell, it has a large surface area. And that's important because with that plasma membrane or cell membrane, we want to get materials into and out of the cell very quickly, and we don't want to waste any time to travel. For instance, if I need to get oxygen into a large cell, it's going to take a lot longer for the oxygen to move outside to inside where it needs to get. Whereas if you have a very small cell, it's going to take less time. And so it's much more efficient to have a small cell and can move materials in and out, and it does not waste any time as far as traveling in there. Now you think about, well, what's the minimum, what's the smallest that we can go? Because you're saying, well, Mr. Stofflet, small, but there has to be a minimum. And that minimum is going to be, well, I gotta have enough space to fit all these parts in. That's the minimum that you can go. So you just can't, there is a limit to the size that you can go. So I showed a picture here. This is maybe a lab activity that you did also in another biology class. You cut out these little pieces of gelatin, and here's a large, and here's a medium, and here's a small. And it was made with a chemical that would react. And so when you soaked it into a beaker, that chemical moved in, and then it reacted with what was in that gelatin. So the gelatin was originally white, and so wherever we see pink, that's where the chemical moved in. So that would be like oxygen or water or other substances moving into that cell. So you can see in the large, it doesn't get to the middle. The medium gets a little bit further, but then in the small cell, we see that it is going to be all the way through. And so that's going to be an advantage to having a small cell size. Another picture I just kind of think about is, take these cubes here, same volume, same size, but take a look at the surface area of this. If I divide this into a whole bunch of little blocks, or in this case I would say a whole bunch of little cells, there's going to be much more surface area here because you've got all of these sides. You've got a six-sided rather than this surface area here. So divide it up, just like our body is divided into the millions of cells, and it's going to be much more efficient at transporting materials for the size. The next page, I'm not going to go through all of these. I don't need to read these to you, but what I'm going to have you do is you're going to make a Quizlet, and we'll talk about these and kind of how they function together. But that's where we're going to start, is to make a Quizlet on the cell parts and functions and what they do. And again, it's not going to be a picture that you have to identify. It's more of, how do they function together. So we're going to move on to endosymbiosis. This is a theory. Prefix endo means inside, and sym means together, or symbiotic relationship means they're living together. So this theory of endosymbiosis is that at one time the mitochondria and the chloroplast were once free-living prokaryotic cells that moved into a eukaryotic cell. So it's thought that at one time this would have been like a mitochondria or a chloroplast at one time. And what this cell did is it moved in to that eukaryotic cell. And so what's the evidence for that? That the mitochondria and the chloroplast both have ribosomes for protein synthesis. Why would you have to have your own ribosomes if not at one time you were living on your own? Because the ribosomes are still present in a eukaryotic cell. So protein synthesis still could occur. So why have your own ribosomes? They have their own DNA, just like a prokaryotic cell. They have double membranes that surround them, and they replicate independently from the cell. So when a cell splits, it's also going to have to make more mitochondria and more chloroplasts, but they do that on their own. So this is an important idea, and we're not sure exactly when it happened, but it would be an advantage to a cell to have mitochondria because they're going to be able to produce a lot more energy. Cells are going to be able to undergo photosynthesis and make their own food in the form of sugar and then use that as an energy source. So there's an advantage, to having these mitochondria in an excess or chloroplasts in excess so that you can produce more energy. And the last thing, which organelles are in animal cells but are not in plant cells? Really, there's only one big one. Lysosomes are going to be found in animal cells because they're going to contain digestive enzymes. So an animal cell takes in foodstuffs and it needs to break it down and digest it, so it contains those enzymes. That's not going to be necessary in a plant cell because plants are going to make their own food. So they are going to take water and carbon dioxide in the chloroplasts and make their own food. So their energy source is actually made in their own cell. They have a central vacuole in a plant cell, so it's going to contain water for the process of photosynthesis, and it's also going to contain a rigid cell wall along the outside. Okay? And so here is, your typical plant cell, your typical animal cell, and comparing those structures. So that's an introduction to the cell, comparing cells, cell organelles, and some other important theories and details about cells.